Okay. Thank you, organizers, for giving me this opportunity. This is really a very small part of the overall project of the Institute of Fundamental Studies to minimize the use of chemical fertilizer in our agriculture and thereby reduce the environmental pollution, particularly by nitrogen fertilizer. This is a global problem, and the FAO UNEP meeting that was held last December last year, resolved to halve the nitrogen use and nitrogen waste globally by 2030. So we are trying to make a small contribution towards that objective. Now, this particular presentation is confined to the use of rhizobial inoculants for bean cultivation in Central Sri Lanka. Bean is a leguminous plant, and when you pull out a bean plant, you will see a lot of nodules in the root system, root nodules. And these have the ability to fix atmospheric nitrogen, which means if you improve nitrogen fixation through the nodules, we will be able to minimize or even do away with addition of nitrogen fertilizer to these crops. So bean is just one crop, but there are so many leguminous crops that are grown in Sri Lanka and all over the world and that are consumed regularly. For example, soybean, mung bean, cowpea, groundnut, lentils, and so on. Now, this study is a part of our efforts to minimize the use of chemical nitrogen fertilizers in agriculture. The overall objective is to contribute towards the Colombo Declaration of 2019 on N management and the UNEP FAO resolution of 2020 to halve chemical nitrogen fertilizer use by 2030. The specific objectives are to formulate and popularize efficient rhizobial inoculants to replace the application of chemical nitrogen fertilizers to legume crops. Now, what are these inoculants? Inoculants are actually the bacteria inside those root nodules which efficiently fix nitrogen. So we select the bacteria and add them artificially into the legume roots so that the nodulation as well as nitrogen fixation by these plants will be improved. And this particular paper will cover the successful application of such an inoculant to vegetable beans. The scientific name is Phaseolus vulgaris. This is the methodology. We isolate the rhizobia from root nodules, select the best strains and grow them in a culture medium in this way in the lab. And once we obtain sufficient pure culture, we multiply them in a broth medium on large semi-mass scale. This is about 6 liters in a 10-liter bottle. And then once the cell number increases to about 10 to the power 8 to 10 to the power 10 cells per milliliter, we inject it into a carrier material. In this particular case, is locally available carrier material, a byproduct of coir industry. It's called coir dust, which is readily available. We sterilize the coir dust, cover it in a polypropylene bag, and inject this inoculant into this, allow it to mature for 7 to 10 days, cover it in a polythene cover, give the instructions of use to the farmer, and finally, depending on the crop, you give a commercially attractive labeling and send it to the market. This particular inoculum I am showing here is for vegetable beans. And then we introduce this to the farmers. This is quite a formidable task because from the time of the Green Revolution in the late 1950s, which was introduced to Sri Lanka in 1960s, for 6 decades, farmers are use of chemical fertilizer, particularly urea. I believe all of us are very familiar with the farmers coming onto the streets if the provision of urea fertilizer is delayed even by a few days. Because today the farmer has got the notion that you cannot grow any crop without addition of urea, which is a complete misnomer. In fact, what we are trying to show is that you can grow crops quite successfully without urea and get the same yields or sometimes even more yields. So that is our mission. Anyway, practically what we are doing here is that one of my assistants, who is a co-author of this paper, Kulasooriya, is showing the farmers how you inoculate bean seeds with our inoculant before it is planted. And then my other research officer, who is also a co-author, Mr. Ekanayaka, is instructing and supervising how the farmers are planting those coated seeds. We intentionally conduct these trials or field testing in this manner with the active participation of the farmers. In this manner, we are sure when this works, farmers' acceptance is almost immediate. It's far better to do this than do all this in research stations and try to instruct them, give them instructions as traditionally done. We have found this approach to be very, very successful. And this is another trial, the organized trial we did. That was farmers' extension work. This was field trial that we conducted in the location Angkumbura, which is about 20 kilometers away from Kandy, where the NIFS is located. And here in a replicated field trial, the layout of the trial, and our research officer, together with the agricultural department, provincial department agricultural personnel, discusses the layout, and bean seeds are planted. Here again, we get the farmers to do the operations so that they get familiar together with our direct guidance. And this is the layout, and here you see the plants are growing at the vegetative, mid-vegetative state. The treatments are marked. And this is the normal bean growth, excellent growth, or luxuriant growth, I would say, that we got with our inoculated bean plants with the farmers, in the farmers' fields. And here is a bountiful harvest that we collected. And in a bean vine like this, you can get about 5 times you can pluck the harvest. And here is a picture of the root nodulation. Here, what is interesting, what I want to explain is that even without any inoculation and without any nitrogen, you can get root nodules formed, because bean plant is what we call a promiscuous plant. It picks up rhizobium from the soil, which is available in the soil. But the difference is, you see, this is the inoculated plant root, this is the uninoculated plant root. In the uninoculated plant, most of the nodules are on the lateral roots. Whereas in the inoculated plants, you see a concentration of nodules, clusters of nodules in the crown roots or roots closer to the taproot. And these nodules closer to the crown or the taproot is far more effective in providing nitrogen than these. These do provide some nitrogen, but this far more, and you get accordingly a better yield with the inoculated one than the uninoculated one. But bean is a promiscuous. If you talk soybean, this won't happen, because soybean, unless inoculated, will not form nodules. But with vegetable bean, it's a different thing. It picks up some rhizobia from the indigenous population in the soil, But when inoculated, it forms a much larger and much higher number of nodules in the crown root. These are the yield results at the farmers' fields in 3 locations of Central Sri Lanka: Anguranketha, Rikilla, and Doragala. And what you see, the green line, the blue line is the control. 2 controls, that is without any fertilizer, urea, without inoculation. And then what you see, the green one is the yield result we get with inoculation. Here you see the green one here. The red one is the one with urea, full recommended fertilizer level of urea. In this particular case, the farmer refused to give us a control plot because he said he'll lose his harvest if nothing is added. So we said, okay, we'll do this. But we clearly see that compared to the control, when we add chemical fertilizer or inoculum, there's an increase. That means there's a response to nitrogen fertilizer by the crop. And here again. But what is interesting is that the inoculated crop has given a marginal increase over the fertilizer. Marginal increase or equal. Now, even if it is equal, which shows that you can replace the urea additions with the inoculant without any reduction in yield. And this is the result with nodulation. When you have the treatment of different levels of fertilizers added, you get 25%, 50% of the recommended fertilizer, 75%, and this is a full recommendation. And this is inoculant without any fertilizer. A low level of fertilizer allows nodulation. As you see, dry weight nodulation, 0.56. But then as you increase the fertilizer levels, nodulation is suppressed. But inoculant without anything, these 2 figures are statistically not different. So they are the same. That means a low level of fertilizer, and in fact, it is recommended sometimes to give a starter dose of nitrogen fertilizer to boost nodulation. This is the yield response curve. That is, we added different levels of nitrogen, that is no nitrogen, 25%, 50%, 75%, 100%. And you see a clear direct correlation. As you increase the levels of nitrogen, you get a higher yield in terms of kilotons per hectare. But what the arrow shows is the performance of the inoculant without fertilizer. And that is not different from not statistically different from that obtained with the highest level of recommended fertilizer. So this more or less confirms or proves without doubt what we got earlier, that inoculants can produce the same yield as the highest level of recommended urea fertilizer. And then another interesting recording that we made with this experiment was, how does the fertilizers and the inoculants affect weed growth? Well, this is a severe problem for any vegetable farmer, any farmer for a short-term crop. And here again, we collected the amount of weed that goes harvested and took the dry weight, recorded the dry weight with no fertilizer, 25%, 50%, 75%, and the full fertilizer. And you see the weed biomass increasing with fertilizer additions naturally, because when you throw broadcast urea to the soil, the nitrogen is available not only to the crop but also to the weeds, and weeds being more aggressive, they will grow and there's an increase. With the inoculant, it is only 5.64%, even less than the control, without any additions, because the nitrogen provided to the plant through inoculation is through the root nodule targeted to the plant. It's not readily available to any other outsiders. Whereas fertilizer, urea or any other fertilizer thrown onto the field will promote beans. So this is another advantage. With inoculant, you get hard, compared to that, reduction in yield in bean cultivation, weed growth with the full fertilizer is 70% reduced when you add the inoculant without fertilizer. And there's another interesting observation that we got. That is, with these central hills, central mountainous regions are undulating mountainous terrain. And most vegetable cultivation, including beans, are done on this kind of huge terraces. And interestingly enough, most of the people who are engaged in these cultivations are housewives and women. All these are women. And when you have to add a fertilizer, recommendation for bean, for instance, is 220 kilograms per hectare of urea. Now, these people have to carry that all the way up to the mountains. Whereas the inoculum we give, as you saw, is just a 100-gram packet for 1 kilogram of bean seeds to be planted. So these people were extremely happy, particularly the women, that they could just take about 10, 20 packets in a polythene bag, walk up instead of carrying kilograms of fertilizer. That's another advantage that we found for the use of inoculants. Now, inferences from this work, this little bit of work done with beans, we have found that rhizobial inoculation could completely replace chemical N-fertilizer applications to bean cultivations in Central Sri Lanka. It also significantly reduces weed growth. It's far more convenient to practice inoculation rather than fertilizer applications. That's taking point view of the people who are working, the cultivators, and particularly the women cultivators. So based on these, I'm happy to conclude that rhizobial inoculants can be recommended to replace chemical N-fertilizer use in bean cultivations without any reduction in crop yields. By extending this technology to all legume crops, the use of chemical N-fertilizer would be reduced substantially, and a significant contribution could be made to achieve the objective of halving nitrogen waste by 50% by the year 2030, as resolved at the FAO UNEP conference December last year. So we can make a contribution toward that. And right now, I should inform you that the National Institute of Fundamental Studies, the Rhizobium project, which is headed by me, has got formulations of this kind of inoculants for soybean, for green gram or mung bean, for groundnut, and for the forage legume clover, which we have field tested and done trials at the Ambevela farm near Horton Plains. And after 2 years of work, we convinced the management that there no need to add any urea to their clover plants because inoculation can do the job. And right now, the NIFS is providing these inoculants for the legume cultivations in Sri Lanka to about 10,000 acres of legume cultivation per year. That is our activity right now. But if the government and other sectors of working on agriculture decides to extend legume cultivation, which is a necessity, we can become self-sufficient in soybean, in mung bean, in groundnut. We can provide inoculants instead of urea fertilizer. And we can clearly give the assurance that This will reduce environmental pollution by nitrogen fertilizer and will contribute significantly for the reduction of environment-related health problems like kidney diseases of uncertain etiology, various cancers, and blue baby syndrome. So there's a lot of contributions we can do if we join hands with the state sector as well as private companies who are interested in the well-being of the population. Needless to say, we are getting the cooperation of the government, that is our ministry is really interested and really helping us with this. And the decision should be to extend the cultivation of nutritious legume crops, which are the poor man's protein in the world. Thank you for your kind attention. Questions?
Professor?
Yes.
Yes. Thank you very much, Professor, for your presentation and your commitment for the conference. So we hope to meet you our next event or before that. So thank you for your spending time with the Gary
Is there
Conference.
any questions or discussions?
No.
Okay, then I can relieve myself because I am planning to leave I'm in Kandy, but I am planning to leave for Colombo within the next 1.5 hours. Can I leave?
Okay. Okay, Professor, you can.
Right. Thank you very much for allowing me to participate in this conference. Thank you.
Okay. Thank you.